One day, a man was walking along when he saw a key on the path. He leant down to pick it up, for he knew this was no ordinary key. This key was in fact the key to the gateway of knowledge that is biology. He walked over to the mysterious portal door and placed the key inside the lock. The man had arrived in the land of biology, where all the most exciting science takes place. The man looked around a while, astounded at this new world of biology, with amazing molecules not yet discovered by man. He walked along for a short way, when all of a sudden, a molecule appeared before him. He took a look at this molecule, and realized at once that this was no ordinary molecule, for this was a very soft and nice molecule. He decided he wanted to touch it and try it on, so he picked it up and decided it looked pretty good on him. Just at this moment, a mysterious stranger arrived. Yo! Whoa! Hey! My name's DNA Helicase. The man turned around to see this new strange man. I'm not a man, I'm an enzyme! And I'm going to tell you about DNA. Did you know that DNA is the origin of all life? It combines bases to form amino acids, which are like totally put together to make polypeptide chains, which are what you and I are made of. The man was excited and eager to learn more about this newfound DNA molecule. So he went over and handed it to his newfound friend, DNA Helicase. Okay, let me tell you what we got here. We got this thing. It's called DNA, deoxyribonucleic acid. And, well, these blue bits, they're like strands, man. And there's two of them. And they go round, which makes them form a helix. And that's important, because these bases have to be protected from all the stuff that's going to mess with them outside. Because they're like code for amino acids, the sequence, which makes everything possible. Now, bases. They pair specifically. A goes with T, and C goes with G. Now, that's specific. Now, if A went with T, and went with C, and went with G, then we'd like too many bases all on one another. That's not going to happen here. Okay, that's what you need to know for now. We'll talk later. The man continued his search for knowledge in the land of biology, when suddenly he had a vision. In the vision, a voice spoke to him, and it told him much, and he learned more than he had ever learned before. He learned that the specific base pairing was caused by certain hydrogen bonds between the bases. For example, the red and green could represent T and A, and they form two hydrogen bonds, which is why they go together. However, the blue and yellow could represent C and G, which form three hydrogen bonds, which is why they pair together. The man woke up from his vision. Man wondered how cells managed to pass on the DNA to the cells that they form when they divided. He decided to sit down and think about this some more. He realized that the DNA had to be broken apart first. To do this, he needed his friend, DNA Helicase. Right, man, I come down like this and I go straight through that DNA, breaking it apart, man, like this. Now then, okay, so I split it up. Now I gotta go now, because, like, the cops are onto me. But, like, my friend DNA Helicase here, who's, like, my brother from another mother, he's gonna, like, tell me with Polymerase, sorry, my friend DNA Polymerase, don't get his name wrong, he'll kill you. And, anyway, he's gonna come and explain a bit more how this all, like, happened. Hello, my name's DNA Polymerase. Now, you see here that DNA strands have been separated. But what happens now? Well, in the nucleus, there are a load of free-roaming base pairs, as you can see here. Now these pair specifically to make a kind of identical thing to this, like this one here. So now, the same thing, but of course these have to be joined together, and that's my job. So once the bases have aligned to each other, I go along and make the ribulose side the phosphate what's the name of that sugar thing? Deoxyribose sugar, that's right! Well done, you biologist people! Anyway, so deoxyribose sugar and phosphate backbone, I make that, once these are all paired, and that makes another DNA molecule. It's called semi-conservative replication, and it's pretty good. Well, I've got to go now. It's like my tea party's on. The man was wondering how DNA turned amino acids into polypeptide chains. To do this, he realized he would need to learn more about two new molecules, mRNA and tRNA. We're going to talk more about mRNA. mRNA is very similar to one single strand of DNA, with the exception being that the T base is replaced with a U base. Don't ask me why. But anyway, so it kind of goes from the nucleus into the cytoplasm. You might be wondering why the DNA doesn't do this. The answer is it's too big to fit out of the nucleus. Kind of like if you were really fat and couldn't get out of your own front door. But anyway, so it copies itself into mRNA. Don't ask me how. But anyway, so that happens. The mRNA is now like a copy of the DNA strand, which is like a gene. Genes code for polypeptides. Polypeptides make proteins. Anyway, that's how it all works. Yeah. So that goes out of the DNA. You got that? You got that? Did you get that? Anyway, so off to tRNA. tRNA, by the way, these are both the same. They're just slightly different for the purposes of running out of materials. Anyway, they have a very specific structure. It's kind of like a clover leaf. Well, yeah, I know. Anyway, clover leaves. Anyway, these clover leaves have anticodons. That's this bit and there. Anyway, these anticodons are important later. We'll find out why soon enough. But they also have an amino acid binding site. That's here and here. Now, let's just add an amino acid. Our amino acids have now got amino acids on the amino acid site. Yeah. Okay, that's enough for now. We'll come back to this a bit later. As the man walked along, pondering biology, a mysterious portal and vortex appeared before him. Goodbye. The man realized he'd learned much of protein synthesis, the last key to the puzzle of biology. Yeah, he decided to share his new knowledge with the viewer. Good eye, mate. Anyway, you see this mRNA molecule here? Well, it's a bit big for what I'm going to show you, so we're going to pretend this is it. So get rid of that, this is mRNA now. This mRNA's come all the way from the nucleus, right? It's now in the cytoplasm where it's met this lovely little ribosome. They get together later and have a nice house in the country. Anyway, mRNA in the ribosome. Got that? Okay, next stage. tRNA! We met that earlier. It's over here. There's two of them. By the way, these are the same thing, they're just made differently. Anyway, so they bind over here, specifically via this anticodon to the codon on the ribosome. Click. Anyway, so the next one does that as well. And they have amino acids on the amino acid carrier site, like that. The amino acids join together, right? Amazing, isn't it? Biological, a wonderful thing. So they've joined together. Now the amino acids are in a chain via polypeptide links. And then the first one goes off because it's lost its amino acid. No point staying here anymore. Anyway, the next one joins along. And that puts its amino acid onto the polypeptide chain, making it bigger. So now we have a polypeptide chain. Wonderful, isn't it? Biologically amazing. Anyway, that's the end now. I hope you've learned a lot about biology. It's going to be great. But there's so much more to learn. And if you didn't find any of this helpful and thought it was a bit distracting, you might need to buy a revision guide, like this one, or ask your teacher for more help, or visit the many biological-related websites online via the internet. Anyway, goodbye from me. It's been wonderful talking to you. Woo!